I am Dr. Khokon Kumar. My Christian name is Dr. David Boone. I am working as assistant professor in the Department of Biochemistry and Molecular Biology in Bangabandhu Sheikh Mujibur Rahman Science and Technology University in Gopalganj, Bangladesh. The title of my discussion is Roles of microRNA-34a in the regulation of Warburg Effect in cancers. Professor Dr. Otto Heinrich Warburg was a German biochemist, physiologist, medical doctor, and Nobel laureate. In 1931, he was awarded Nobel Prize in Physiology for his discovery of nature and mode of action of respiratory enzyme. Dr. Otto Heinrich Warburg investigated the metabolism of tumors and the respiration of cells, particularly of cancer cells. One of his famous observations was extremely high level of lactate production in the tumor and cancer cells. It is to mention here that the end product of glycolysis is pyruvate. Glycolysis takes place in the cytoplasm of a cell. Then pyruvate is metabolized in the mitochondria for the production of usable energy ATP for the cells. But in anaerobic condition, pyruvate is converted to lactate in normal cells. In tumor and cancer cells, high levels of lactate are produced even in the presence of sufficient oxygen. Dr. Otto Heinrich Warburg tried to explain the observation in a way while low mitochondrial respiration or respiratory damage in the mitochondria or respiratory impairment contributes in the high lactate production in tumor and cancer cells. He tried to introduce these respiratory damages or impairment as the common driving forces in the carcinogenic process for almost all cancer types, which is known as Warburg effect in cancer biology. Warburg effect is well known and widely discussed and investigated even today by all the researchers and scientists related to cancer biology throughout the world. Previously, lactate was known only as the metabolic end product. But increasingly, it is evident now that lactate is related with diverse types of functions in tumors and in cancers, and in the regulations of different pathophysiological aspects in the body of a cancer patient. Lactate is now known to be involved in the epigenetic regulation of gene expression. Lactylation of histone proteins in the lysine residues plays important roles in the regulation of gene expression, including oncogenes. Lactate was once believed as the metabolic waste, but it is known today that lactate is used as an important fuel for energy production and even can be used by the Krebs cycle. Lactate plays important roles in almost all the states of carcinogenesis, from the tumor formation to metastasis. Lactate plays important roles in the tumor and cancer microenvironments. Lactate is now known to play important roles in the modulation of immunity. Immune cells infiltration is almost always found within the tumors and cancers. But lactate suppresses the immunity inside the tumors and cancers. Lactate helps the immune evasion process for cancer cells. Lactate acts as negative regulator for both innate and adaptive immunity. Lactic acid affects the intracellular and extracellular pH, which is an important regulator of cancerous growth. Lactate is also known to be involved with other functions too. Lactate plays important roles in the angiogenesis, wound healing, and in other regenerative processes, memory formation, and others. Rapid growth and proliferation is characteristics of early embryonic development. The presence of high lactate is found in the early embryos. High lactate also works as an important factor for the stem cells, which show a similar growth pattern like cancer cells or cancer stem cells. At present, a huge research reports are seen to find out the explanation of Warburg effect in the genomic and molecular levels. Suitable permanent blockage in the respiratory process in the mitochondria may be capable to result the Warburg effect in cancer cells. But permanent blockages may not be an obligatory or essential choice in this case. Because muscle cells in the human body are capable to shift metabolic mood from aerobic and anaerobic glycolysis depending on the conditions they experiences. So it should be likely that there may exist some mechanisms in the cancer cells capable to change the metabolic mood in a reversible manner, even though the reversible practices are not used by the tumor and cancer cells. At present, some mechanisms which regulate the Warburg effect in cancer cells have been discovered. Khokon, Kumar, Dutta et al. From the School of Medicine, Kyoto University, Japan, published an article in Cancer Science in 2007 with the title Association of microRNA-34a overexpression with proliferation is cell type dependent. We investigated the expression of microRNA-34a in a huge number of human cancer samples using in situ hybridization approaches. We found high expression of microRNA-34a in almost 50% of cases. It is to mention here that microRNA-34a expression is related with p53 in a little bit complex fashion, and abnormalities with p53 is found in almost 50% of the human cancers. But the main focus of the study was centered with the renal cell carcinoma of rats, where we found high level of expression of microRNA-34a in cancer cells. The high level of microRNA-34a expression in renal cell carcinoma has also been reported by other groups recently. We also checked the expression of microRNA-34a in almost all cell types of the body of normal rats. We found low level of microRNA-34a expression in the major metabolizing cells like hepatocytes, renal tubular cells, and myocardial cells. But we found high expression of microRNA-34a in the neurons of the brain, which largely depends on glucose as energy source. Otto Warburg published an article in the Journal of Science in February 1956 with the title On the Origin of Cancer Cells. He reported that some non-neoplastic tissues like brain, retina, kidney medulla, and intestinal mucosa consume moderate to high level of oxygen and operate high degrees of glycolytic activities similar to tumor and cancer cells. Depending on the similarity in high microRNA-34a, we try to hypothesize a commonality in glucose metabolism between cancer and neural cells in the brain. Rong Zhang et al. Published an article in January 2016 in the American Journal of Cancer Research with the title HPV-E6 p53 mediated downregulation of microRNA-34a inhibits Warburg effect through targeting LDHA in cervical cancer. They did a gain of function study with 12 microRNAs that are frequently found with downregulated expression in the cervical cancers. microRNA-34a was one of them. microRNA-34a directly suppresses lactate dehydrogenase A, LDHA, and inhibits Warburg effect in cervical cancers. Lactate dehydrogenase A, LDHA, catalyzes the last step of the glycolytic process. In the study, they used two different cell lines, HeLa and Shinya. They found that knockdown of oncoprotein E6, expression of human papillomavirus in Shinya and HeLa cells by siRNAs lead to an increased protein level of p53, decreased level of microRNA-34a, as well as reduced Warburg effect. In our study in the School of Medicine, Kyoto University, Japan, we investigated 177 human cancer samples, which was collected from South Korea. We included 10 samples for cervical cancers, and all of them were squamous cell carcinoma. We used in situ detection approach for microRNA-34a. The pathological grading was 10% of the samples were negative for microRNA-34a. 50% of the samples were 1 positive, that means almost negative. And 20% of the samples were 2 positive. And the rest 20% were 3 positive. The results suggest that there may exhibit different types of expression and involvement for a particular gene in cancers originated from the same cellular type in the body. About 60% of the samples of cancers used in our study in Japan supported the previously discussed findings published in 2016 in the American Journal of Cancer Research. Till today, several other protein coding genes have been reported as important regulators of Warburg effect in different cancers. For example, p53, hypoxia-inducible factor 1, HIP1, TP53 induced glycolysis and apoptosis regulator, TIGER, pyruvate kinase M2 isoform, PKM2, phosphofructokinase PFK, and others. Eva Slebakova et al. Published an article in 2017 in the Journal of Cell Death and Disease with the title Alternative Mechanisms of microRNA-34a Regulation in Cancer. They reported the multiplicity of the target of messenger RNAs for microRNA-34a. microRNA-34a suppresses about 700 messenger RNA transcripts in tumor. Someday we may find some mechanisms in tumors and cancers where high levels of microRNA-34a can even positively induce the Warburg effect. Finally, I would like to wish happy, prosperous, and dignified life for you everybody. Thank you very much.